Great. So I would like to also echo Carolyn's point. It really is a great honor to be invited by the grad students. And I always never say never, right? That's what I've learned, but pretty much never have ever turned down an invitation by grad students. So I think it's really an honor and something that we as professors need to do to show encouragement to graduate students to move forward. Anyway, what I'd like to do in the next 35, 40 minutes is to discuss some research that we've been doing in my laboratory. And I'm going to use the example of, maybe it works, maybe it doesn't. I guess it doesn't work. What's that? It's on the side? Ah, has to be put on first, right? So essentially, I'm going to talk about tomatoes, but essentially you can change that for any crop that you want. Okay? The strategies that we're trying to develop for making disease-resistant plants, you'll see can be applied to almost anything. What I'm going to talk about today is a disease that I've collaborated with Bob Stahl and Jeff Jones and others at the University of Florida over the last 20 years, trying to really understand the control of the bacterial spot disease of pepper and tomato. And you'll become aware of it just a second. Just as an introduction, because I know that many people aren't plant pathologists here, I just want to make a couple of points for this particular introductory slide. So as we understand right now, the plant innate immune system is pretty complex. And it's kind of interesting, if you're not in the field, everybody thinks, oh, everything's known about plant immunity and everything's known. But I can tell you, there's so many unanswered questions about plant immunity. We know a lot, but we don't know a lot of real specific mechanisms that are involved in resistance. So there's still an amazing amount of information to uncover. But what I want to point out here is at least two layers of the plant immune system. And we'll get to a third one in a moment. But one of the first layers of the plant immune system is called PTI. And P stands for PAMP. So there's a lot of acronyms in this whole game. So PAMP stands for pathogen-associated molecular patterns, or some people call them MAMPs, microbial-associated molecular patterns. These are essentially components that are on all microbes and that can be recognized by plants. Okay, examples would be like flagella, chitin, LPS, all kinds of common components that microbes basically present to plants that can be recognized. And oftentimes they're recognized by what I call pattern recognition receptors. So these are receptors that essentially have a leucine-rich repeat that goes outside the cell, and inside they have a kinase domain that's involved in signaling. So for instance, here we have a gene that's called FLS2, we'll get to that in a moment. It recognizes flagellin, and that establishes an immune system, kind of a basal layer of immune system. Now, what happened basically during the evolution of plants and pathogens, what we found out is that all pathogens, including insects, nematodes, they all have a very common feature. They all inject proteins as virulence proteins into the host cell. And we were starting to uncover what many of these particular effector proteins do. They either modulate or inhibit the immune system. And often they're targeting the PAMP-triggered immune system. Okay, so you have these organisms like bacteria, nematodes, aphids, fungi, or oomycetes such as Phytophthora that can inject these effectors in, and oftentimes they can inhibit this pathway. But they do many other things. Okay, this is just for an example in this particular case. But also now plants evolved, they had to have a mechanism to be able to overcome this particular virulence components of these effectors. And so plants basically evolved what basically is breeders as plant resistance genes. And the major class of plant resistance genes are called NLRs. These stand for nucleotide binding leucine-rich repeat proteins. And they kind of come in two major flavors. They come in essentially with an N-terminal, what's called the coil domain, and then it has a nucleotide binding site and a leucine-rich repeat. Or it actually has a toll interleukin-1 receptor N-terminal domain and a nucleotide binding and leucine-rich repeat. These are kinds of, now what's interesting, the monocots don't have chair NBS LRRs, which is quite interesting, but the dicots do. So we're seeing a dichotomy in the types of these, evolution of these immune receptors. So these immune receptors either directly or indirectly recognize these effectors, and these would trigger the effector-triggered immune system, which is more robust, which is often facilitated by a hypersensitive or cell death response in the host. So you have these two layers that are playing, but there are many other layers and other intricacies that are going on, but I just wanted to bring this up because this will become important as we go on. So what is the challenge for molecular plant pathology? So I would say, despite many seminal advances over the last 30 years in the elucidation of the molecular basis of host resistance and pathogenicity of phytopathogens, there are very few examples where the knowledge has been translated to solve practical problems in agriculture for disease control. So if we could deploy GMOs right now, we could solve many of the disease-resistant problems out there right now. But of course, we've heard about the public not accepting GMOs as an approach. Obviously, it has, interestingly enough, before everything got going, but this basically saved the papaya industry in Hawaii, basically making RNAi for papaya mosaic virus. But the main problem still is the public acceptance of GMOs, and then also the cost of deregulation, okay? So it's a problem. It's not going to go away, but again, I agree, we have to put this in a context. So we'll talk about genome editing and see how that's going to be perceived by the public also. So the bottom line is, what we're really interested in doing is to take a multi-pronged approach to make disease-resistant plants. Because many of you know, and you've heard the stories many times, is that if you breed for disease resistance, and usually plant breeders like to deal with dominant genes because they can select for it in the first generation. And it's just a matter of time before the pathogen, you put a selection pressure on the pathogen is going to evade that host resistance. It's going to be called a defeated resistance gene. And the pathogen now is going to become virulent on that previously resistant host. And this has gone on for many, many years in cycles of plant breeding for disease resistance. So how are we going to solve this problem? So one approach that we're doing is that we're interested in combining many of the 3 layers of plant disease resistance for making a more durable type resistance. Whether we can actually make durable resistance remains to be seen, because pathogens are smart. They always find ways to get around. Anytime you put selection pressure on a microbe, they'll find ways around it. But hopefully we can try to out-trick them. So one thing we're interested in doing is using pattern recognition receptors. And I'll give you an example where we can transfer the EFR receptor into tomato that significantly reduces the multiplication of a Xanthomonas and a Ralstodia bacterial pathogens. Can we actually make stacks of disease resistance genes? That essentially, so if the pathogen mutates one of its effectors, and it can overcome resistance, but if you have a multitude of resistance genes, you would have to have a whole series of mutations that would come to override those particular pathogens. And then what I'm quite interested in these days is really identifying plant susceptibility loci. So it turns out there are genes in plants that the pathogen actually induces that actually makes it favorable for the pathogen to actually grow. So if you can go in and start to manipulate those, and the other caveat is that they don't have some kind of developmental pleiotropic effect, you can actually use these to combine with all these multiple layers of disease resistance. So pattern recognition receptors, again, this is just a nice review by Cyril Zipfel. And we talked about FLS2, EFR, which recognizes this translation factor from bacteria. And essentially, what the experiment was is that we took a susceptible tomato plant and we transformed the EFR receptor from Arabidopsis in the tomato. And lo and behold, when it did that, it actually recognized the effectors from several different pathogens. And so essentially this paper here in Nature Biotech several years ago showed that you can transfer a pattern recognition receptor that confers broad spectrum bacterial resistance, both to Xanthomonas and actually to Ralstodia. Okay, but this would come under the aegis of a GMO, okay? Would this would ever be accepted remains to be seen. But there are ways of potentially genome editing these types of things also. We can talk about that also. So here, this is quite, in this particular case, you can see that it's, EFR plants are quite resistant. Here's another thing that we can do now, which I think is extremely exciting, is that using the power of genomics, because what we can do now is we have the power to go out and to be able to ascertain in real time in the field the genomes of pathogens, okay? Now, it's obviously more challenging for the larger pathogen populations out there. For bacterial pathogens, though, it's quite easy. These days, we can sequence a bacterial genome for about $27, okay? Not counting the grad student postdoc time. That's basically the cost. You can multiplex these and really sequence. They're only 6 megabases in size genomes. So what we've started to do is to actually collaborate with Bob Stahl, and the idea is to go out and survey fields and to kind of do it in multiple year times, and to really understand what is the genome of the pathogen out in the field? What's the natural variation that is out in the field? And more importantly, we have a computational pipeline that we can ascertain what effector proteins are in those pathogens. And so how do those effector proteins, are they changing? Are they under selection? You can look at allelic differences between them. So you can generate a lot of data and really start to look at the pathogen population. So this is what you could call almost an effector-mediated plant breeding. And one real lifetime is that the Dutch, right now, actually, what they do is they go out and analyze Phytophthora in the field, in potato fields, looking to see whether an effector is present, that's the corresponding resistance genes in the potato variety. And if it's there, they won't spray with chemicals. But if the effector is mutated or changed, they'll actually spray. So they use it as a management tool to actually understand the genomics of the pathogen in the field. So I think it's quite a interesting thing. So this is all kind of published stuff right now, but essentially we've collected various pathogens, and so we're interested in trying to understand the core types of things. This was published a year ago in Frontiers of Microbiology. So it's basically the phylogenomics of field strains of these pathogens. You can then start to look at all the different types of effectors. I'm not going to go, don't worry about the names up here, just these are effectors. But what we did is we found a common core set of effectors that were all in these particular pathogens. So what can you do with this information? Well, what you can start to do is that you can try to find corresponding effectors that can recognize these particular, NLR proteins that can recognize these effectors. And the idea is that if we can find ones that recognize the majority of these, we can stack these up in a plant that confer resistance. And more recently, this one, SOC2, we have identified a resistance gene. You'll see that we have one for ABRBS2, and we're seeking to do this. Well, it turns out that these effectors, you can inject these into all kinds of different plants, and you can induce a hypersensitive cell response, cell death response, which suggests that there's an NLR protein. And we're not going to get into the basics of non-host resistance here, but probably part of non-host resistance can be explained by recognition of effectors from these particular pathogens. So here's a kind of an example that we've done, that this has been around for a long time. This bacterial spot disease in tomato caused by Xanthomonas perforans. So 92% of Florida, about 40% of the US growing area is infected by this. Chemical control is ineffective. People have tried to use copper sprays, and you can imagine copper resistance has appeared. Even though they still use it, if you go to Florida, you would actually see tomato fields blue, but it actually has absolutely no use whatsoever. In tomato, there really isn't any effective genetic resistance that's really available. And Xanthomonas perforans is now the dominant strain. So a long time ago, we cloned a gene from pepper called BS2 for bacterial spot number 2. And it turns out that this particular gene, oops, we're going the wrong way here. Yeah, okay, here we go. This particular gene comes from a wild species of pepper. Oops, I gotta go back. There we go. So BS2 comes from Capsicum chuckoense, and it was introduced in the Capsicum annuum, and it recognizes an effector gene called ABRBS2 that is conserved in all Xanthomonas strains out there that affect all kinds of plants. So what we did, and these days, this is a long time ago now, that Tom Tye back in 1999, this shows you how long this field's been cranking along for a long time, is that we took this gene, and we first proposed this experiment and said, no, this will never work. You never take a pepper gene and put it in a tomato plant and actually be effective. But it turns out it's extremely effective. And we are able to actually get very good resistance. We've done 6 years of field trials in Florida now, where we can actually get very good resistance. And here it was published a couple of years ago. But what's more important is the total and extra large marketable hybrids were actually came out in this particular field trial. So this is an extremely valuable gene, but the tomato growers said they would not use it because of acceptance of growers. And here we try to make the point, and I think there's another way to try to convince people about GMOs, is that copper is a very nasty chemical compound. It's actually found in the packing sheds in Florida. It could be cancer causing, it accumulates in the soil, it's nasty. So here we have an example. Maybe we could use a particular genetic approach that would reduce chemicals that are nasty for the environment. So that's, I guess, another example that one you could use to explain why you would want to use genetics as opposed to chemicals. Okay? So what's interesting, even more bizarre, is that BS2 is in every single pepper that you actually eat. Okay? So it's obviously very safe. And so whether we're going to be able to, and I'll show you an example of what we're trying to do for genome editing with this at the end, which hopefully could be more accepted, but maybe not. So it confers resistance, and it's really important because it confers a yield benefit, and it also has this resistance removes the need for environmentally hazardous chemicals. So let's go into CRISPR-Cas9 now. So it's pretty amazing. If you think about it, CRISPR-Cas9 was published in 2012, okay? And what we've seen right now is a revolution. I used to, every Friday, go onto PubMed and do a search for CRISPR. Well, now you get about 150. You can't even read the abstracts that are coming up right now. To stay up on top of this literature is almost impossible. Every person is using, this is the PCR of plant breeding. I'll show you in a moment here. So basically, it's a technology, and it's extremely potent, and it's going to really make a difference in how we proceed, I'd say, with plant breeding in general. So what we've been doing is to construct gene mutations employing CRISPR-Cas9, and the easiest way to make mutations is to call them non-homologous end joining. So this is where you make a double-stranded cut in DNA, you allow the repair system to mismatch, and you get a mutation. Okay, this happens very frequently, and you can get plants homozygous usually in the first generation. What everybody wants to do is to perform allelic exchanges to introduce precise amino acid changes in existing genes, or to put epitope tags onto naturally occurring particular proteins in plants so they can study the function of them in a natural setting. Okay, this is a lot more difficult. This is a low frequency event, and there are many people around the world now trying to establish procedures to actually increase homologous recombination and to actually get precise allelic exchanges. This is once this is cracked, which I think will be. This is also a problem in mammalian cell cultures too. It's not unique to plants. Homologous recombination is a low frequency event. It happens. So what you have to do sometimes is produce thousands of plants where you can get 3 or 4%. You screen through all those. Okay. So, but it's a lot of work, and it costs. If you had to regenerate 1,500 maize plants, it's expensive, right? So you can think about it. The other thing that Jonathan Jones, who's a long-term colleague of mine at the Sainsbury lab in Norwich, we've been talking about what we call knock-in breeding. So knock-in breeding is basically strategies to introduce characterized disease-resistant genes into elite inbred parents of tomato to eliminate linkage drag. Still, as you probably know, that many resistance genes come from wild sexually compatible species of a particular plant. Breeders have bred these particular resistance genes in. And then there's a whole series of recurrent selection and backcrossing, and oftentimes it's by chance whether you can find a recombinant from the stuff that you don't want in there. Okay, the phenomenon of linkage drag is real. It's still real even by tomato breeders. So being able to get the gene. So what if you could then just take the gene itself from the wild species and introduce it precisely into the same location that it normally might have gone in by genetic crossing? And this is one of the APHIS guidelines that are currently up for review right now. This would be considered a non-GMO. A gene that can be normally crossed in from a sexually compatible species, but CRISPRed into a particular location would not be considered a GMO. Okay? You can think about it now as you take your elite inbreds, and you start adding all the, and this doesn't have to be resistance genes, it could be any trait that you're interested in, that you want to put into there. So you don't have to go through this long series of backcrossing. I think this is going to be really the power of CRISPR in genome editing, is really just the next breeding tool that's available to actually perform elite inbred constructions. Let me tell you another approach that we're doing. So people have taken mutagenesis approaches to try to identify genes that are important for susceptibility. And a colleague of mine, Guido Vandenack in Utrecht University in the Netherlands, did this very nice experiment with Arabidopsis and this downy mildew pathogen, which is an oomycete called Hyloperonospora arabidopsidus, or HPA, if you want in this particular case. And what they did is a forward genetic screen. They took this particular plant, they mutagenized it, and they looked for resistant plants. Okay? And they identified a gene called DMR6, which for downy mildew resistance number 6, and it belongs to a superfamily of 2 oxoglutarate iron-dependent oxygenases. Okay? And they're implicated in secondary metabolism, biosynthesis of signaling molecules, and so on and so forth. It's also thought that this might be, and there's some controversy in the literature, whether this actually may be a salicylate hydroxylase, which would activate salicylate in this particular case. So, downy mildew resistant gene 6, interestingly enough, not only gave resistance to HPA, it started giving broad spectrum resistance to Pseudomonas syringae, Phytophthora capsicum. And so it was quite interesting. And what a lot of this was related to, most likely, and you'll see, is that it correlates with increased salicylate levels. But oftentimes when you increase salicylate levels, you often get stunted or really small dwarf plants. But in this particular case, there doesn't really seem to be a major off-type with this particular mutant. There may be a reason for that. So it's interesting that DMR6 is activated only during pathogen infection in cells neighboring pathogen growth. So this is, in this particular case, they have a GUS promoter being driven by the DMR6 promoter driving GUS activity, and you only see activity around invading pathogens. So this may be why you don't see these off-types. So you can take DMR6 and you can do a phylogenetic analysis amongst all kinds of different plants. And of course, we were interested in tomato in this particular case, and you can see there are 2 candidates here for tomato in this particular case. And so we wanted to ask the question, could we then, taking the known orthologous genes in tomato, can we knock those genes out and can we basically mimic the type of disease resistance in tomato that was seen in Arabidopsis? So the first thing we did was to just take available literature out there. We knew that the DMR6 gene is naturally induced after pathogen infection, and we showed that to be the case with Pseudomonas syringae, Xanthomonas, and also Phytophthora. Okay? You can identify the genes similar to what we just saw. You can identify exons. You can use all kinds of programs similar to what that we just saw, that you can identify CRISPR sites by identifying the PAM site. And essentially, these initial experiments where we used agrobacterium, where we had Cas9, and I'll talk about that in a moment, and we used various guide RNAs to target the Cas9 to a specific site, where it then would recognize double-stranded DNA and make a cut, and through non-homologous end joining would make a mutation at that particular location. So we heard about Cas9. It's really quite a remarkable enzyme. So Cas9, the major feature is it has multiple domains on it. One is it has 2 nuclease domains, so it can produce double-stranded double-stranded nicks. But more importantly, what it is, it has a helicase domain. Basically, when it gets double-stranded DNA, it unwinds it and allows it to go into it, where it searches out the guide RNA to the complementary DNA that's going to make that particular cut. And so what happens is, at the majority of the time, you get this process called non-homologous end joining, and you'll get these mutations. But if you can use homology to direct, if you want homologous recombination, which happens at a much lower frequency, you can actually do precise homologous recombination events. So this is kind of a nice little demonstration of the structure here. So if you look at Cas9 protein, it's the white blobs here. The guide RNA is in orange, and the target strand is in the dark blue, and the non-target strand of the DNA is the light blue. So the Cas9 comes in, it opens up the particular DNA molecule, and here you can see essentially where the PAM site is going to be, and it goes in, and here it cuts the particular double-stranded DNA, and you get this particular one. To make a long story short, we are able to get mutations, and what's remarkable, at least in tomato and several other plants, is that you can get a fairly high percentage of either homozygous or biallelic mutations in the first generation, okay? So that's quite remarkable. And we now know that in polyploid plants, even in wheat, which is a hexaploid, you can get homozygous mutations for the MLO gene in wheat, actually, in some of the first generations. So it's really quite remarkable that you're really able to get efficient gene knockouts and get homozygous or either biallelic, so you might get two different alleles, but they'll be both knockouts at the same locus. So what you can do, at least in plants, is that you can now self those plants, because Agrobacterium is basically in there, in the Cas9 is actually in there, there's a hemizygote in that particular case. And essentially you can segregate the Cas9 away, but the mutation you can remain. So you go through a couple of generations and you get the mutation and you segregate everything else away. So the only thing you have is a mutation now. This also, under the current proposed guidelines, would not be considered a GMO also. Okay, all you have is the particular mutations. Now, the other thing you can do is that you can go and search whether you have off-target effects. That would be obviously important, but at least with plants itself or easily outcross, you can actually segregate away off types if you needed to. So what we found is that just like in Arabidopsis, we found really good resistance to Pseudomonas syringae, which causes bacterial speck disease. We saw resistance to Xanthomonas gardneri, which is one of the major pathogens of bacterial spot. And look at this, we saw resistance to powdery mildew. This is even more amazing. So there's a powdery mildew called Iridium neolicopersica in this particular case. And here's the control plant, and here's the resistant plant. You can see that, phenotypically they look pretty similar. And obviously these plants are pretty clean, and those are diseased with powdery mildew. Seem to work pretty well with Phytophthora capsicum also in this particular case. It's not an all or none thing, but it's greatly reduced for disease in this particular case. So this is kind of an interesting, and these types of diseases, if I don't know in what happens in Iowa, but I know that even in California, if I grow tomatoes in my front garden, it's in Berkeley, so it's kind of cooler. I have to grow it up against the stucco part of my house on the south-facing side of it and stuff like that. But I'll get late blight that will actually wipe out my crop at the end of the year. So it may be that putting this particular gene into, let's say, heirloom tomatoes, I think personally, I think a great idea to get people to accept this type of technology is to release it to home gardeners, where they can actually say, "Oh, look at this, I can grow my tomatoes without using any chemicals. And it's only a mutation for this particular gene." And they would brag to their next neighbor, say, "Hey, you should be getting this right here and use this." This, I think, because look, at home gardeners love these, they like to tinker with these types of things. And especially if you could put them in heirloom varieties that taste great and everything else. So I think this is another way you could potentially communicate to the public this technology is safe and it could actually be very beneficial to you personally. I would also add to what we heard about GMO acceptance. Of course, the initial ones were all farmer input traits. I think if we started developing tomatoes that tasted great in February, all kinds of things, peaches, apples, anything that tastes better, that's better for you nutritionally. Think about GMOs in medicine. Every medicine you take, most of it is from a GMO. No one cares because it has a personal benefit. I think selling it to people as a personal benefit is really important. That's my own personal opinion in some of these. That's the way I tell people. Here's another example that we're trying to use genome editing. So plant breeders don't like usually to use recessive resistance, because especially in a tomato, which is a hybrid seed variety, most hybrid seeds, the trait usually only comes in from one of the parents. So if you actually had to put a recessive gene in both parents, it's difficult. But with CRISPR, you can easily put it into the elite inbreds that you're going to make the hybrid seed. So in collaboration with Jeff Jones and also groups in Hungary, we were able to clone a recessive gene to bacterial spot. And it's an interesting gene, and it's called BS5, and here it's just in this isogenic line. And what's interesting is that it's a non-hypersensitive resistance. It doesn't really give any symptoms, and the bacterial growth is lower. We have no idea what the mechanism is, but we wanted to actually identify what is the gene that's involved in recessive resistance to this particular bacterial pathogen. So, in order to do this, you saw the previous talk. I'm just going to quickly show you. This was done the old-fashioned way by map-based cloning. These days, you could actually do by map-based sequencing, where you could bulk the segregants and just sequence and find SNPs that would be close. You could then use CRISPRs to identify potential candidates and knock them out. But this was done the old-fashioned way, where essentially you had a resistant plant, a susceptible plant, 4,662 F2 guys were, there's three things important in map-based cloning: recombinants, recombinants, and recombinants to your closest markers. How far can you narrow down the particular locus that you want between the closest markers on either side? Once you establish that, then you can identify the physical region of DNA that spans those markers, and your gene should be in that particular region. So that's what happened here. It was narrowed down to these. You don't need to worry about the details there, but essentially we were able to narrow it down to a particular gene. It turns out it's in pepper. It's called the BS5 gene. There's a very similar gene that's transcribed in the opposite direction. What's interesting is it's a bizarre gene. There's nothing you can tell you really what it is. There are other genes that are out there, and you'll show you in a moment here. But the nature of the mutation essentially was a 2-leucine C-terminal in-frame deletion that gave rise to this resistance. Okay. Some other experiments we've done is that we have an assay where we can look at the delivery of effector proteins, and we can see that the effector delivery is greatly lowered in this, but we don't know whether it's a specific effect or it's a pleiotropic effect. This is actually in this particular case, this is this gene here. When you line it up, there's some differences. The nice, interesting thing is you can get a gene from pepper, and you know that in the solanaceous plants, like in the Poaceae, there's a lot of synteny between the various chromosomes. So if you find a gene in pepper, it's most likely it's going to be an orthologous gene in tomato. And lo and behold, we were able to find a gene here, as it was very similar. And what this gene is, if you want, it's highly similar to a neighboring gene. It's 92 amino acids. It has this 5 amino acid repeats, and it's a cysteine-rich terminus. And it's part of this PGYRP gene family. And where there are mutations in Arabidopsis, it's thought to be involved in development or response to stress. Okay, so now you've found a gene, you can probably use it, but you don't understand how it works. Okay, most likely it's hypothesized to be essentially in the membrane, but we haven't determined that yet. So just kind of in the final parts here, what we've been also doing is we talked about this whole idea of knock-in breeding. And this is, I think, really, going to be the major influence of CRISPR-Cas9 in breeding, is that here you have a high-yielding disease-susceptible plant, here you have a low-yielding disease-resistant plant, and then you're interested in introgressing it in, okay, the phenomenon of linkage drag. So now, the tomato genome is quite well annotated. You don't have to worry about it here, but these are all the resistance genes that have been identified. And there are even more that have been identified, and there's still more to be identified. And in an unrelated project, we have a grant where we're collaborating with Jonathan Jones, and we're looking at all the wild species of tomato and looking at all the resistance genes that are in those species. There's a particular technology called RenSeq, which essentially is an exome capture of all the NLR proteins, and we can then basically annotate them and do phylogenetic analysis and look at particular orthologous genes that are in, and we were interested in all the wild species of tomato. Coming back to the BS2 gene, just as I told you about the BS5 gene, there's an orthologous gene in tomato of the pepper Capsicum chacoense gene that's about 71% identical. Okay? So this again, this is just a lineup right here. So one thought is, can we take the Capsicum chacoense open breeding frame, and can we insert it behind the native promoter of the BS2 orthologous gene in tomato, such that it would then be driven in the exact location of that particular orthologous gene? So we don't have the results yet, but we are using CRISPR-Cas9 to make essentially double cuts, to be able to try to use homologous recombination to insert the pepper open reading frame behind the promoter and the terminator of the orthologous gene in tomato. Okay. Will that be regulated or unregulated? Currently, the guideline probably would still be regulated as a GMO, but hopefully we could make an argument that this gene is being eaten by everybody, and if you're just putting it in the tomato, what's the difference? And it has a great benefit for the environment and for human health. So basically, the summary here is we're interested in combining a multi-pronged approach for disease resistance. Okay, we don't want to rely just on disease resistance genes. We'd like to combine into plants pattern recognition receptors, stacked resistance genes, and also susceptibility genes or recessive resistance. So the idea would be is that the recessive resistance would lower the growth of the pathogens, which would then lower the probability that the pathogen would be selecting for mutants that would overcome the resistance genes. And the idea is that can we slow that process down such that we can then have a long-lasting disease resistance phenomenon by using a multi-pronged approach. We're trying to use various processes to perform homologous recombination to create novel recessive genes for disease resistance. We're introducing and characterizing genes into elite inbreds to expedite plant breeding and eliminate linkage drag. And then we're also, as we said, introducing a combination of stacked R genes that recognize multiple independent core effectors. So remember, understanding to manage disease resistance is not just understanding the host. You have to know the pathogen just as much. The pathogen is essential because understanding what you're trying to resist against and how variable that pathogen is and what kind of effectors are present in those pathogens out in the field is extremely important. And we have the technology to do that now. And so I think if we start to deploy genome sequencing or field pathogenomics, if you want to call it, give it any name you want, but really understanding the pathogen populations in natural fields and what's going on there, even as you change new varieties and how that goes to the particular pathogens that come around. So also, CRISPR-Cas9, we talked about it in several approaches here. I guess another great tool in plant breeding is that when you have QTLs and you have candidate genes, CRISPR-Cas9 can immediately go in and look for a function for those. So I think that's going to be a really important aspect of plant breeding also. So I'd just like to give this slide. This is, someone asked me, was Berkeley on the coast? And I said, not quite on the coast, but it overlooks the coast. So essentially, this is from the Berkeley campus, and you look at San Francisco and the Golden Gate Bridge. And I'd just like to recognize the Two Blades Foundation, which has done a lot of the work, our Jorge Kiss and Zoltan Szabo, and our Hungarian collaborators, NSF, USDA, Sally Miller at Ohio State, Jeff Jones in Florida, and people from my laboratory. So I'll stop there. Be happy to answer any questions. I think they want you to get up there, Roger. You have to, unfortunately, you have to stand up.
I'm interested in this 2 amino acid frame deletion. Do you have any idea what the mechanism that changes the function of that protein?
We have really no idea. We've used VIGS, virus-induced gene silencing, to knock that out in the plants. If you make, I think, if you make a null mutation, I think the plants are sick. So there's something about that protein, and we think it's a plasma membrane inserted protein, but we really don't have any idea. This is part of trying to get at the function of genes. You can clone genes, but figuring out their biochemical function, if it's not obvious, it's not an easy task. But it's something we're very interested in trying to get together. Yeah. everybody gets faced with these types of problems. Question?
Thank you. It's a very nice presentation. I just have a lot of questions related to this following the first question about these 2 amino acid changing, how about the causal gene has more than 2 amino acid changing? How are you going to move to the next step?
So well, that's a good question, because here's another thing you can do with CRISPRs. So if you think about it, if you do non-homologous end joining, you can make an allelic series of genes, okay? And so that's what we're also trying to accumulate now. So then you could then make different types of mutations and ask whether they are altered or you need that specific one or anything else. But it's in a way of making allelic variants. That's something that just randomly, and people are using this right now, I can tell you, to find really interesting phenotypes for all kinds of traits in plants, architecture traits, all kinds of flowering time. They're essentially just totally going in and making promoter mutations in all kinds of genes. You can find upregulation, downregulation. You can make a whole allelic series of genes that have really interesting phenotypes. But we don't know the answer to your specific question, but we're trying to make new alleles.
Great presentation. So my question is, like, all this CRISPR-Cas9 technology we use basically for different genes. But being breeder, we focus more on the quantitative traits, which are governed by so many genes and everything. So how do you think like this technology will help us in future for those kind of traits?
Well, as I said, I think that one way is that if you have QTLs, and oftentimes people don't know what their QTLs really are, right? So if you can get it down to some candidates, you can, let's say you have 6 candidates marked, and then you can just go in and knock them out individually or pairs or whatever. So it becomes a functional genomics tool to really go in and knock out those genes, and you see a phenotype. I think that's where it's going to really help in those particular cases. Another question?
You talked about CRISPR-Cas being non-GMO and the cost of deregulation. Do you have any idea what the regulation bodies like the EPA, FDA, what their requirements are going to be?
So there is APHIS, which the USDA has published a whole set of proposed guidelines, and a simple mutation caused by genome editing with CRISPR-Cas9 that doesn't have Cas9 left in the plant. So there's several ways to do it. You can do it with agro, and then you can segregate the gene away. Or what most people are trying to do are DNA-free CRISPR, where you just use the ribonucleoprotein, which is purified Cas9 protein, in vitro complex with guide RNA that you make in the laboratory, and then you use particle bombardment to shoot that in. That becomes very efficient in mutations also. So there's basically no DNA involved. There's protein and RNA. It makes mutation, and that doesn't get inserted into the genome. That's probably the cleanest way to make that. And then secondly, it's proposed that if you have genes that are normally genetically crossed in with sexually compatible species, that introducing those genes in by this technology would also not be regulated. So that's what's proposed. And I think they're supposed to have some resolution by July, I think is the time frame for where, I think it's 6 months it has to be published in the National Register. But of course, with Trump, we don't have to worry about everything. Nothing will be regulated anymore. GMOs will be totally free.
In a company, like when you stack all the multiple mutation genes together, is it possible that, like in the future, someday you can create like super resistant bacteria? What is that possible?
Right. So the question is, if we start to make all these alterations in the plant, are we going to select for some super strain of bacteria or a super strain of pathogen? It's always a possibility. And there's other ways of thinking about how to deploy different resistances. You can do them in kind of different combinations in the field, not have everything genetically identical to one another. So you can start to use some epidemiology and other things to put in it. So there are many ways to proceed with it. It's certainly something to think about, and it's important to think about. I agree.
So, Brian, it seems it must be the tomato, elite minds of tomato tolerate going into tissue culture and go through all the steps that you have to go through to subject them either to transformation or even CRISPR-Cas9 genome editing. Do you have some idea about how other crops, because right now that's the hang up, you heard it
in an earlier talk, we said, well, B73 is not transformable, so it's not transformable, it's just that it won't tolerate all this.
Maybe you can ask our next speaker, because Pioneer has cracked this with Wuschel and Baby Boom, where they supposedly have genotype independent transformation in maize. I think it's probably lower, but I think it's greatly enhanced and it's much faster, and it doesn't really produce as many epigenetic changes. We can certainly find out from our next speaker whether that's the case or not.
And so I guess that was the question.
So I think there are people who are trying to crack that to really, because right now there's always a few varieties that can work, right? And then,
It's easy, you're still stuck with that process.
right. So essentially then, can you crack it with these types of transcription factors that seem to allow things to move much quicker from immature embryos into full plants? But it's tricky how you add them and how you remove them and everything else. But you guys are right up the road from the experts. You should ask them.
Well, I thought we expanded the list of lines, but that still doesn't fix the problem, which is we need elite lines, not some.
Right. Well, again, we can ask our next speaker. I don't know how many elite lines of maize, but my understanding is that it's pretty good, at least in maize. And I know that my understanding too, wheat might be very similar to it, that they've cracked it also in wheat. In most monocots, I think they've cracked this with Baby Boom and Wuschel. It's more difficult in dicots, but at least in monocots, I think this has been cracked. Anyway, time will tell. Yeah, let's put it this way, it's becoming infinitely better.
Maybe better than others.
Yeah.
Still got a little ways to come.
Well, yeah, that's an important thing. How do we actually get that technology into the academic sector? I think that's really important to do that. It's kind of crazy just to have a certain company have that technology and not the rest of the brains of the world trying to do it, because it's a limiting factor. Right? Everybody has ideas what they want to do, but if it's too difficult or too expensive to do it, it really limits what can be done. Something to think about. Okay, any more? Thanks very much. I guess is there a break then?
Yes, so we will have our second offer section and the coffee break. So we'll see you again at a little bit before 4 to receive the provided our guest speaker.